and welcome to today's webinar on cell death detection tools, finding clues in morphology and biology. This talk will explore the importance of cell death in maintaining cell health. I will discuss major types of cell death, highlighting the key morphological and biochemical hallmarks associated with each type of cell death, and also discuss the different products and services that Cayman offers to study cell death pathways. Cell death occurs for both physiological and pathological reasons. Varying modes of death are either programmed to safely remove healthy cells that are no longer useful, or to stimulate an inflammatory response to clear cells that are useful but no longer healthy. The first type of cell death I would like to talk about is apoptosis. Apoptosis is an evolutionary conserved cell death pathway that is responsible for the programmed culling of cells during normal eukaryotic development and maintenance of homeostasis in organisms. It allows for elimination of damaged, infected, or cancerous cells and plays an essential role in the development and maintenance of a healthy immune system. Cells that undergo apoptosis go through an orderly, non-inflammatory process. They shrink and develop bubble-like protrusions or blebs on their surface. The DNA in the nucleus gets chopped up into small pieces, and some organelles of the cell, such as the ER, break down into fragments. In the end, the entire cell splits up into small chunks, each neatly enclosed in a package of membrane. In terms of biochemical hallmarks of apoptosis, we see activation of effector caspases 3 and 7, which then initiate and execute apoptotic degradation. There is also caspase-triggered opening of plasma membrane channel pannexin-1, leading to loss of intracellular ATP. Furthermore, normal cells display phosphatidylserine in the inner leaflet of the plasma membrane, but apoptotic cells display phosphatidylserine on the outer leaflet of the membrane, facilitating phagocytic recognition. In addition to the activation of caspases 3 and 7 by initiator caspases, there is also activation of other pro-apoptotic proteins that lead to permeabilization of the mitochondrial outer membrane and decrease in potential of the proton gradient across inner membrane of mitochondria, causing release of even more apoptotic proteins like cytochrome C. Cayman offers annexin V assays that can be used to probe for exposure of phosphatidylserine on the plasma membrane. Cayman also offers DNA stains that can be used to indicate membrane permeability, as well as mitochondrial membrane potential assays, multiplex assays, caspase activity assays, activators and inhibitors, BCL2 inhibitors, p53 antibodies, assays and activators, as well as antibodies against programmed cell death, RIP-like interacting Clark kinase, and apoptosis repressor with CARD to study apoptosis. The apoptosis assays that Cayman offers include the early apoptosis detection assay kit as well as the multi-parameter apoptosis assay kit. These multiplexed assays include multiple markers for various stages of apoptosis to allow for quantitative segmentation of total sample population into distinct phases at the single cell level. The next type of cell death I would like to talk about is necrosis. Necrosis has been classically defined as an unprogrammed form of cell death that occurs in response to infection, physical or chemical insult, or lack of blood supply. Necrosis was primarily thought to be passive when cells are irreparably damaged by external forces, but recent data has uncovered programmed form of necrosis that I'll go into more detail in later slides. Typical morphological hallmarks of necrosis include cell swelling and plasma membrane rupture, which lead to activation of inflammatory responses. Cayman offers membrane-impermeable probes like DAPI, propidium iodide, and 7-aminoactinomycin D, as well as the cell-permeable inhibitor of necrosis, Necrox-5. Moving to an active, programmed form of necrosis, also known as necroptosis. Necroptosis is induced by ligand binding to the TNF family death domain receptors. In recent years, necroptosis has been recognized as an important viral defense mechanism. Cells undergoing necroptosis depict cell swelling and plasma membrane breakdown similar to necrosis. However, at the molecular level, during necroptosis, there is activation of RIPK and subsequent activation of MLKL. Cells undergoing necroptosis expose phosphatidylserine on the outer leaflet of plasma membrane similar to the apoptotic cells. When the plasma membrane of these cells start breaking down, they release danger-associated molecular patterns or DAMPs, triggering inflammation. In addition to annexin V assays used to probe for phosphatidylserine exposure, Cayman also offers MLKL and RIPK inhibitors to help researchers study necroptosis. There is another type of programmed necrosis called pyroptosis. Pyroptosis is triggered by pro-inflammatory signals including DAMPs and PAMs and subsequent activation of caspase-1. Cells undergoing pyroptosis have similar morphological changes as cells undergoing necrosis and necroptosis, that is, cell swelling and plasma membrane rupture. However, at the molecular level, pyroptosis requires caspase activation and cleavage of gasdermin D at the C-terminal domain to release the membrane pore-forming gasdermin D N-terminal domain. Pores formed through gasdermin D N-terminal domain promote the release of pro-inflammatory contents of the cell. Cayman offers caspase-1 antibody, caspase-1 inhibitor screening assay kit, as well as caspase inhibitors to study pyroptosis. Here is more information on the caspase-1 inhibitor screening assay kit. This is a fluorometric assay that can be used to identify novel inhibitors of human caspase-1. The assay kit includes potent and reversible caspase-1 inhibitor as a positive control. Yet another form of regulated cell death is ferroptosis. Ferroptosis is a newly identified form of non-apoptotic caspase and necrosome independent form of cell death. Ferroptosis involves iron-dependent lipid peroxide accumulation. Cells undergoing ferroptosis depict cell swelling and increased mitochondrial membrane density. The most notable features of ferroptosis at the molecular level include glutathione peroxidase 4 and ferroptosis suppressor protein 1 inactivation. There is iron-dependent ROS accumulation, and with the defective lipid peroxide repair system, there is accumulation of lipid peroxides. Cayman offers FSP1 and GPX4 inhibitors and screening assays, as well as lipid ROS detection probes and assays. Cayman offers antioxidants, iron chelators, iron oxidizers, lipoxygenases, oxidized phospholipids, as well as inhibitors for cysteine glutamate exchange transporter. Here is some more information on the FSP1 and the GPX4 inhibitor screening assay kits that Cayman offers. The FSP1 fluorescent inhibitor screening assay kit can be used to identify novel inhibitors of human FSP1, which is a negative regulator of the ferroptosis pathway. The assay measures the reduction of rasazurin by FSP1 into resarufin. The kit includes the recombinant human FSP1 as well as the positive control FSP1 inhibitor iFSP1. The GPX4 inhibitor screening assay kit can be used to identify novel inhibitors of human GPX4, which is also a negative regulator of the ferroptosis pathway. This is a colorimetric assay kit that measures GPX4 activity indirectly by a coupled reaction with glutathione reductase. And the kit includes recombinant human GPX4 and positive control GPX4 inhibitor ML162. The last type of cell death I would like to talk about is NETosis. NETosis is a neutrophil-dependent cell death. When neutrophils are activated in the presence of inflammatory stimuli, there is increased NADPH oxidase activity resulting in large amounts of superoxide production, subsequently leading to secretion of large web-like DNA structures decorated with histones and cytotoxic proteins called neutrophil extracellular traps or NETs. Morphologically, neutrophils undergoing NETosis display chromatin decondensation, nuclear envelope disaggregation, nucleoplasm homogeneity, cell membrane rupture followed by chromatin expulsion. At the molecular level, there is increased ROS production and PAD4 activity leading to citrullination of histone H3 and the release of myeloperoxidase, neutrophil elastase, and defensin in NETs. Cayman offers citrullinated histone H3 ELISA kit, NETosis assay kit, and the imaging kit, as well as PAD4 inhibitor screening assays, enzymes, antibodies, and inhibitors. Here is a little bit more detail on the NETosis specific assays. The citrullinated histone H3 ELISA quantifies the most specific marker of NETosis, which is the citrullinated histone H3. Citrullinated histone H3 is produced most exclusively during NET formation. The assay has been validated in mouse bone marrow leukocyte and serum, as well as human plasma and serum. The NETosis assay kit detects elastase release from stimulated neutrophils, and this does not depend on the DNA component of NETs. This assay is compatible with any source of NET-producing cells. In addition to these, Cayman also has the NETosis imaging assay kit. Cayman also offers other resources like guides, wall charts, and scientific posters on cell death. You will find articles, application notes, and brochures on cell death on the Cayman website. In addition to these, Cayman also offers services in various research areas. Thank you.